8:05, right? Was that? Okay. All right, let's begin. Okay, so in essence, we have 2 sessions together today. Both sessions are related, but hopefully we'll have a nice break in between to refresh you. And what I'm going to do is something that is simple and highlights primarily key points that I would like for you all to understand at a fundamental level, obviously, so that you can carry forward to whatever system that you are dealing with. Okay, so focus on, in essence, the key points, more on fundamentals. Information is important, obviously. You need to know quite a bit about proteins and things of that sort. But if you understand the fundamentals, then you should be able to use it fairly easily. So, in general, obviously carbohydrates mediate whatever that they mediate, biological response, physiological response, by interacting for most part with proteins. Theoretically, they can interact with any macromolecule. It doesn't have to be proteins. We are simply using proteins as an example because there are a large number of proteins that interact with carbohydrates, glycans. And overall, we can classify all the proteins that interact with glycans into 2 major categories. Okay, one of them the first one is lectins, and the second one are gag binding proteins. So there are some nice similarities and there are differences. Okay, so that's what we'll try to focus on. Now, lectins were discovered more than 100 years ago, and over a period of time we have come to realize that we can classify lectins in a large, good number of classes. For example, L-type lectins or C-type lectins, R-type lectins, I-type lectins, and these are all lectins. Lectins, for most part, are thought to recognize neutral carbohydrates, neutral glycans. Okay, lectins recognize neutral glycans. That doesn't mean they don't absolutely recognize Gags. Some of them do. But it's just that the predominant molecules that these recognize are carbohydrates. L-type lectin, the letter L is basically derived from leguminous plants, and that's where they actually started being discovered. C-type lectins are essentially calcium-dependent lectins. R-type lectins are ricin-like lectins. So there is something 3-dimensional in terms of residues and all commonality amongst this specific subgroup, which is why we classify all of these into these distinct categories. I-type lectins are immunoglobulin-like lectins, molecules, lectins that recognize sialic acid are more specifically called Siglecs. P-type lectins are mannose-6-phosphate recognizing lectins, galactins are beta-gal recognizing lectins, and there are more lectins than what we can write here. There is no universal classification of lectins. People tend to put these lectins into various different classes according to what they like, according to the similarities that they have. Now, gag binding proteins, we have discussed a few of these. For example, there are serpins. Antithrombin, which we discussed yesterday, is a serpin. Serpin, the word serpin stands for serine protease inhibitor. Okay, so these are inhibitors of serine proteases of a specific type. So you would see, for example, BPTI, bovine pancreatic trypsin inhibitor, is also a serine protease inhibitor, but it's not called serpin. Okay? BPTI is not called serpin. Likewise, there are several other proteins. These are proteins that inhibit serpins. But the word serpins is used for a specific class of molecules that have three-dimensional similarity. We saw the structure of antithrombin, and I didn't really point out at that time the number of helices and beta sheets, typically 3 beta sheets, 9 helices, and plus some additional features. The commonality of this arranged in that particular manner generates a serpin. And a serpin has a specific mechanism, and we saw that mechanism yesterday in a little bit cursory manner. But antithrombin is one of the serpins, and then heparin cofactor 2 is another serpin, which is also our major focus. Likewise, Kubi pointed out a serpin called PCI, protein C inhibitor, is a serpin. There is a serpin called, this is Kubi. He's lost perhaps. Hello? Hey Kubi, I'm teaching. Okay, bye. Okay, so we have protease nexin 1 called PN1. Protease nexin 1 also binds heparin. Just as heparin cofactor 2 primarily binds dermatan sulfate. Protease nexin 1 binds heparin, and protein nexin 1 is present in the brain, and heparin accelerates protease nexin 1's reaction with thrombin more than 1,000-fold. Okay, and the mechanism is still being figured out. ZPI or ZPI, PI stands for protease inhibitor, is also a serpin, and it binds heparin, and heparin accelerates its reaction, not to the extent of 1,000-fold, but approximately 50 to 100-fold. There are enzymes that bind Gags, and we have seen thrombin, factor 10A, factor 9, factor 11A, factor 9A, factor 7A yesterday. Chemokines bind Gags, and Dr. Rajaratnam is going to talk about a lot of this stuff pretty soon, tomorrow morning. And obviously, we have discussed a little bit about growth factors and their binding and the receptors and the complex formation. So, and there are a number of growth factors, and these are just very, very few. Obviously, as you all probably know, because you have been students of Gags for quite a bit, so there are a large number of other proteins that also are called Gag binding proteins, which are not really listed here. So, here's a simple comparison, and I'm taking this from the book that I plan to give to each of, I've given to some of you, and this is from Essentials of Glycobiology, page 376. And it's a very nice table that summarizes a lot of information about Gags binding, glycans binding to proteins. So lectins and Gag binding proteins, if you look at shared evolutionary origins, lectins have significant similarity. Gag binding proteins have essentially minimal similarity. They are scattered all over the place. Okay? And therein lies the challenge. Structural features, because of this, obviously the structural features are fairly similar. Here, because of absence of this, there is essentially no structural similarity. If you look at the amino acid residues that are involved in binding, then often there are significant, for most part, identical amino acid residues are involved. Group of amino acid residues are involved in recognition. Even if it is mannose versus glucose, for most part, similar residues are involved in recognizing these hydroxy groups. Here, It's basically a patch of basic amino acid residues. And we don't really know exactly what governs high affinity interaction. If you look at the type of glycans that are recognized, then obviously lectins recognize neutral sugars. Some of them do recognize sulfated, and lectins also recognize sialic acid containing molecules, as Kubi said, terminal residue. And these recognize Gags, sulfated Gags. For most part, the recognition domain is it sequences at the ends of the glycan chain. So the last terminal residue is the one that is typically recognized. Here, the sequences is actually more than just a single residue for most part. And you know that you can have as long as a pentasaccharide, as we saw yesterday. But what I'm going to show you in a short while is that even longer chains may be recognized. The binding sites are really long. If you look at the specificity of glycans, lectins are highly stereospecific. You have phenomenal specificity. Lectins can recognize a particular lectin can recognize a glucose but not mannose, a glucose but not galactose, and there is a single hydroxyl stereochemical change, which I have pointed out earlier also. In this particular case, for most part, most GAG binding proteins don't recognize too many, don't have very high specificity. In terms of affinity, for most part, lectins have very low affinity for individual monosaccharides. Individual monosaccharides have very poor affinity, as low as a millimolar. Okay? Yet, they have a mechanism for specific recognition. That's the beauty about it. They have a mechanism for specific recognition. For most part, GAG binding proteins recognize their sequences with moderate to high affinity. Moderate being micromolar, high being nanomolar. There are very few proteins that recognize GAG with really high affinity.
You're using the word avidity.
Yeah.
So does that mean specificity or affinity?
Yeah, I'm going to classify what is the difference between. Where did you see that? Oh, very good point. So this avidity and affinity are two different things. Avidity is derived from affinity. I'm going to talk about that in just a little while. Okay. So valency of binding sites, for most part, these are multivalent. So you have several domains that recognize these residues. Okay. Which is called multivalent or bidentate or tetradentate. For most part, these are monovalent. Okay, and obviously there are subgroups, a large number of subgroups. For example, here we have heparan sulfate, chondroitin sulfate, dermatan sulfate binding proteins, and the recognition within that group is fairly good. One of them we have already seen. Heparin cofactor 2, for example, recognizes more dermatan sulfate, less heparan sulfate. Okay. So I think it's a fairly concise description of the entire group of proteins in a nutshell, to be able to reasonably know as to what you can expect. Go ahead.
So if you remove the sulfate groups from GAG, do more lectins tend to bind them?
If you desulfate last residue, if it is galactose, yeah, you might recognize, sure. Completely desulfate, yeah. Mm-hmm. Okay, so a little bit about lectins, discovered more than 100 years ago, primarily through a simple phenomena that you can actually see in hand, which is called, for most part, precipitation. Those days it was used agglutination. Cells would bind each other pretty strongly, and they would precipitate, they would agglutinate. So these were RBCs, red in color, and therefore that process was called hemagglutination. And people wondered as to, why do these things, group together and precipitate. So once in a while a scientist had a strong solution of glucose, and hemagglutination did not really occur, indicating that glucose for most part would suppress it. And that's how logic came around that, well, there might be some proteins that are recognizing glucose. And later on, that actually became a very simple assay. If you really want to find out the specificity of interaction, you basically take a solution of several different sugars and you try a glutination assay. The one that prevents it, you would hopefully identify the sugar. The concentration of glucose, for example, has to be fairly high because the affinity is not that high. So it is also obviously a method for detecting specificity if you have a library of carbohydrates. This group of molecules, earlier they used to be called agglutinins. Later on, they were renamed lectins, and the word lectin arises from the Latin legere, which means to select. So it's basically a highly selective process, and that's a very characteristic feature about lectins. Every plant species has lectin. In fact, lectins are present in essentially entire kingdom, all life forms. They are most abundant, as I said, in leguminous plants. In viruses, you have heard quite a lot, which is the reason I put it here. These days there is a lot of discussion about H1N1 and H5N1 and things of that sort. So there are viruses, and and Kubi also talked about this a little bit. So there are viruses that specifically bind alpha-2,6, whereas human influenza viruses, where the bird influenza viruses bind sialic acid alpha-2,3. And that's a small change. And therefore there is a significant talk about whether the birds can actually jump to humans. In fact, there is a big discussion right now going on whether to publish a paper in Nature and Science. There are two papers that are being discussed. The final approval has just come through, and they have said that we should publish it. Appropriate mutations have now, in the virus genome, have now made that transfer easier. So the thinking was that if you publish this, then it can get into wrong hands, and they could engineer a virus that could be a bio threat, things of that sort. So there's a lot of discussion going on, and that has to do obviously with aerosolization of the virus and transfer. But in general, for most part, humans were protected because of this recognition feature, that they would not really work. Yeah, so that's really interesting. Did somebody say something? Okay. Now, likewise, bacteria also has a lot of lectins. In fact, for bacteria, these can have dual effects. They can be beneficial and they can be toxic. All right. So I'm selecting very simple examples to simply illustrate the principle. So they can be beneficial. It's not that you can have pathogenic things all the time. For example, some of the molecules could actually facilitate colonization. And this colonization is dependent on a specific structure. And that's a glycan structure. All right? So in our large intestine, in a large intestine, we typically express that domain, gal alpha 1,4, gal beta 1,4, and glucose beta ceramide. And this particular domain is not expressed in the small intestine. And this is the domain that is recognized by certain bacteria. So what we have is we have selective colonization of these bacteria, and it's a good thing. At the same time, there are certain bacteria that can be toxic, that induces pathogenic event. So cholera toxin, for example, binds GM1 ganglioside receptor, and this occurs through specific glycan recognition. That leads to all problems. So you can have both pathogenic, and it's a beautiful field to really investigate as to how glycan recognition is really important. Glycan recognition, as you all know, we talked about in the first slide itself, is really important for many of the immune responses that we have. ABO blood is basically dependent on glycans, which I'm not listing here. Okay, animals. Animals have several types of lectins, and what I'm highlighting is one specific type, asialoglycoprotein receptor. Okay, asialoglycoprotein receptor. So you all have been very quiet, and I want to ask you a little bit simple question. What does that mean? What do you think would that receptor do? Asialoglycoprotein receptor. What would it do? Don't look at that. Don't look at that. Don't read it. What would it do? What do you think it would do? What's that? Yes, very good. Asialo. So, not sialic acid. So it would basically recognize glycans that do not have sialic acid. So what typically happens is sialic acid, as he said, puts a cap. And for most part, what happens, in fact, very recent discovery, very recent investigation. Why doesn't it not work? Very recent discovery is that platelets, and platelets are important, right? Platelets are really important for good clotting to really work. So platelets, for example, have on its membrane certain glycoproteins that have glycans with a sialic acid. Okay? And platelets have a life of their own, and what happens is that sialic acid is clipped. That sets off a signal, and asialoglycoprotein receptor present in liver and there's also another receptor that says that, okay, this platelet now has to be taken off the system. So it's a beautiful clearance mechanism, which means, and to give you an idea in terms of the application, so how would you use it to apply? Basically, for people who have a problem and we want to inject platelets, there has to be platelet storage, right? Invariably, platelet storage leads to increase in the synthesis of neuraminidase. Neuraminidase is basically one that clips off platelets, and therefore stored platelets cannot be stored for a prolonged period of time. So the idea, therefore, is that if we know the mechanism as to how asialoglycoprotein is playing a role, then what do? We basically prevent deacylylation, okay, and thereby we extend the life of platelets so that storage becomes much easier. A very simple application of the basic biochemistry. Yeah, are you all with me? I know you were all disturbed, something happened outside, so. Okay, all right, let's move on. We've already spent half an hour on just 3 slides. Okay, so if you really look at cells, and Kubi tried to give you an idea about this too, as to the overall dimension of the glycan body on a protein, that's what he was trying to do. What I'm showing you here is on a cell. If you really look at the cell surface, okay, simply the cell surface. So you have all heard about the cell membrane, the bilayer, and there are proteins, integral, and GPI anchor, and those are proteins, and there is cholesterol, and that's not a glycan, and things of that sort. That's what you all heard of, right? But if you really look at the cell surface, what is this, TEM? I have not written it. I think most probably this is TEM image. So if you really look at the surface, the entire surface, what do things outside see? Basically carbohydrates, okay, and that is typically called Glycocalyx, okay, Glycocalyx. And this is what, and each individual's Glycocalyx is unique. So we have our own way of responding to whatever things that come around. Okay, so that's pretty interesting. All right. So now let's just talk about few principles of neutral sugar recognition, okay, by proteins, by lectins for most part. And I'm giving you general principles, very general principles. So if you really look at a monosaccharide binding to a lectin, it's usually weak, very weak, sometimes high micromolar to millimolar. And yet, as I said, that there is considerable stereospecificity and very good recognition. How does that arise? That arises by the protein, in this particular case, CON-A, forming multimeric species. So when you have a multimer, you actually engineer several binding sites. And when you have several binding sites, and you have Glycocalyx that I talked about, what typically happens is that these residues, multiple copies of these residues are exposed outside. So you have the multimer recognizing, for example, these two simultaneously. And that enhances the affinity dramatically. Yeah, are you all with me? Right? That enhances the dramatically affinity. For most part, majority of lectins, CON-A is the prominent lectin. We talked about CON-A even in chromatography, right? Affinity chromatography. So because of that, the affinity increases. In fact, if you really look at the increase in affinity, it's pretty phenomenal. Typically, it's more than 100-fold. And this is called oligomerization or clustering. Not many people use oligomerization. Most people use clustering. And clustering is from the glycan perspective. Oligomerization is from the protein perspective.
Are these induced oligomerization by the...
They could be. But under native conditions also, and these are all crystal forms, they are oligomers. I don't really know much about the study in vitro about monomer oligomer equilibria. But for most part, majority of them are dimers, tetramers, and higher order oligomer systems. So it raises the affinity greater than 100-fold. And in one particular case, it raises the affinity more than 10,000-fold, which is the basis for high specificity. I keep repeating this because it is really, really important. The recognition is really stereospecific. Across the board, we've already talked about 2,3 versus 2,6. We have talked about glucose versus galactose. CON-A, for example, recognizes glucose and mannose. And the difference is primarily at position number 2. Okay, and I think I have a structure. Although it recognizes glucose and mannose, the way in which it recognizes is different. Okay, so likewise, we have, for example, N-acetylnuraminic acid recognition versus, as you have seen, that the acetyl can actually have a hydroxy, and that difference. Go ahead.
It's just an observation about this. Just a comment. I think CON-A is very immunogenic to humans, but typically it's always used as a model system, although it has no consequence in humans. And everybody makes.
There's a lot of thermodynamic kinetic studies. So much study has gone. It's an easier system. It's the first system. A lot of other factors that go into it. Sure. Good point. Very good point. Okay. So monosaccharide recognition typically arises from organization, obviously, of the binding sites such that, for most part, nearly all hydroxy groups on the sugar are recognized. Okay, nearly all. Because if you really want to figure out a difference of 1, you can't just recognize that particular site. You have to basically recognize everything around. And that is engineered either directly by the side chain or the main chain groups of the protein, or through water. Okay, so water plays a fairly important role. Here, if you really look at, so this is a structure of one particular lectin, and this is a structure of the second lectin. This is mannose versus this is galactose. Okay? This is mannose versus galactose. And in essence, the binding site corresponding residues are very similar. The binding site corresponding residues are very similar. So the binding site, although the binding site is significantly similar for two different lectins, the way in which it recognizes these residues is different. O4, O3 is being recognized by this group, whereas O3 alone is being recognized by this group. So you can see a twist which is engineered in the recognition principle, which generates the high affinity. Okay? So overall, the differences in geometry, preferential geometry, is the reason for specificity. And the lectins don't create an alternative binding site, a new binding site, in order to recognize a different monosaccharide. That's interesting. That's really neat in how these things. So there's very minor structural differences that engineer this high level of specificity. Point that I should mention. So there is hydrogen bonding that I mentioned, and then recognition also involves metal ion coordination. So there are metal ions that play a role. For most part, you might have one metal ion, you might have two metal ions. There are van der Waals forces, and obviously there is water-mediated hydrogen bonding, or actually water release. Lectins can have several metals, as I said, one or two. And the metals, or metal ions, actually are for most part involved in folding of appropriate amino acid residues so that the binding site can form. So metal ions can be directly involved. The metal ions can actually also recognize water, but also importantly, metal ions may be involved in appropriate folding, so that creation of a binding site. Believe it or not, hydrophobic interactions might also play a role. Okay? Hydrophobic interactions may also play a role. So here, how does it come about? So here we are presenting, this is the glycan. All right? This is the glycan. This glycan in this particular crystal structure has been at the reducing end, has been modified by an aromatic ring. But the specificity remains the same. So it turns out that the bottom part of this glycan is oriented in such a manner that it forms hydrophobic interactions with tyrosine 12, the aromatic ring. So even hydrophobic groups may be involved, and the phenomena is very simple. Certain sugars, certain monosaccharides have, in essence, for most part, all polar groups organized on a particular face, and the rest of the face, for example, the bottom part here, is fairly hydrophobic, and that generates specificity. So if you can imagine, if this hydroxy group is placed here, then you would have not good interaction, and that engineers specificity. Okay, water molecules. Water molecules are obviously very important. Sugars are, in essence, completely surrounded by water molecules. And binding, the binding site itself may also have a lot of water molecules. So when the glycan binds, it might actually release some water molecules. Okay. At the same time, some of the water molecules might actually be involved in the binding site. So you have both phenomena going on. You have an entropic phenomena, the release of water molecules actually help binding, or you might have an enthalpic phenomena where water molecules are actually involved in the interaction. And there is always competition. Let me ask you a question. What is the energy of a hydrogen bond? What is the energy that a single hydrogen bond can provide?
5 kilocalories.
Approximately 5 kilocalories, yeah. When would it provide 5 kilocalories? Okay, what is the definition of a hydrogen bond?
So electronegative atom, hydrogen, and bond with another electronegative atom.
Right, so it's a sandwich, 2 electronegative atoms and then 2 strongly electronegative atoms and then a hydrogen in between. So it's 3 points and has to be linear, right? Has to be linear. So when the 3 point, 3 atoms are absolutely linear, the angle is 180 degrees, is when it actually gives 5 kilocalories per mole, approximately 5 kilocalories per mole. But in nature we have varying degrees. So it's not necessary that you have 180 degrees. It can be from 150 degrees to 180 degrees. You can have that kind of a variation. And that's still considered strong. Anything less might have hydrogen bonding character. So you might actually get 1, 2, or 5 kilocalories per mole. Here is a simple illustration of how much of an increase in affinity can actually arise by few kilocalories per mole, which is in essence net 1 hydrogen bond, okay, for most part. So if you have a delta delta G0, right? What is delta delta G0? Change in free energy, right? Change in free energy. You know the equation for delta G0 and affinity or equilibrium constant? What is the equation? Delta G0 is equal to, you all know this, come on, minus ln K, but there are also some other 2 factors, RT, minus RT ln K. So if you use that equation, so there's going to be K1 or K new and K old, okay, right? You know how I'm converting all this, so that's what I'm trying to say. So ln minus RT ln K old before the formation of hydrogen bond, for example, and after the formation of hydrogen bond, if you subtract delta delta G is, delta delta G0 is delta G new minus delta G old is what I'm trying to say. Yeah? And because it is ln RK, so it will become K new minus K old, so it becomes a ratio. Becomes that. Are you all with me? Okay? If you write it, you will be able to figure that out. It's not difficult. Okay? Right? So if delta delta G0 is approximately 2 kilocalories per mole, small hydrogen bond like character, 1 net, not necessarily just 1, net, then you will see an increase in affinity by increase or decrease in affinity depending on whether it's formed or not, of approximately 30-fold. That's a 30-fold change. Okay? If you have a stronger net hydrogen bond, let's say 4 kilocalories per mole, it's 800-fold increase in affinity. And if you have 6 kilocalories per mole, and we are not straying too far away from net 1 hydrogen bond, it's approximately 24,000-fold increase in affinity. So you very quickly go from a millimolar to 10,000-fold, millimolar to low micromolar. Yeah? Low micromolar. Very quickly you go if you start from millimolar. Yeah? Which is also illustrated somewhere here in terms of the release of 1 water molecule. Okay? All right. So let's quickly move on. We already spent 45 minutes on lectin, so I want to spend little time on Gags, okay, or gag binding proteins. And so once again here there's a partial listing. I think we have been discussing this at length, so I don't want to go into much detail. But a big category of gag binding proteins are actually present on enveloped viruses. Okay, and this is true for herpes simplex, HIV, dengue. Lots of viruses have glycoproteins, and these glycoproteins recognize gags, and there is a reason for that. It's a beautiful, nature's beautiful mechanism of infection. Okay. So another big category is lipid binding proteins. Lots of apolipoproteins bind gags. In fact, and this is a beautiful paper that if you're interested you should check out. It's bioinformatic analysis of something that they say is a heparin interactome, trying to analyze proteins in our body that bind to heparin based on bioinformatic analysis. And guess what? There are 435 proteins that they suggest actually interact with heparin. Now we don't really know whether all of these interactions are physiologically relevant. We don't know that. Whether all of these interactions are specific or non-specific, or whether we can exploit them for pharmaceutical purposes. But in essence, you can imagine the number of proteins that we can work with, right? It's a huge number of proteins that we can actually work with. Can I go forward? You're all very silent, so. All right, so this is an aspect that we were discussing a little bit yesterday, and I completely forgot that I was going to talk about it in this particular session. But here is the crystal structure of heparin. Is it heparin? Yeah. Here is the crystal structure of heparin binding to factor 9A. Okay? Binding to factor 9A. And superimposed on this structure is the structure of heparin binding to thrombin. That's basically indicated by white lines. So I'll point out for you. This is thrombin. This is not thrombin. This is heparin binding to thrombin orientation. And this is heparin binding to factor 9A orientation. Thrombin and factor 9A are homologous serine proteases. Thrombin, 10A, 9A, 7A, 11A, this is 9A, 11A are all homologous. The heparin binding site is homologous. The positive charges, there are few differences, absolutely, there are differences, but it's basically homologous. But it's essentially perpendicular arrangement. Completely different arrangement. And this is crystallography. Okay? So,
How similar the structures are?
how similar were the protein structures?
Yes.
Fairly superimposable. Fairly superimposable. And this is really true. This site is for most part called, in thrombin is called exosite 2. And for most part, I think people have not started calling it exosite 2 in 9A or 11A. For most part, it's a homologous site. And you can see the blue is basically all positive charge. Do I say that? Yeah, strongly electropositive. Thrombin is known to be non-specific. Factor 9A, for most part, I think at the present time, people think that, yeah, it should be non-specific. Yet the orientation is completely different. Okay, can I go forward? Yeah? I think it's a simple point, so I think we should be hopefully done in 10 minutes. All right. Gags, and here I'm showing heparin. Because of their negative charge, the entire negative charge and the repulsion that arises from the negative charge is basically a helix. Okay? So, in essence, we have a helical arrangement, and shown here are model structures. So this is not really experimental structure, but this is model structure in which one of the conformation of the iduronic acid is 1C4 across the board, which is A, and the second conformation is 2SO across the board in B. So if this is uniformly 2SO, uniformly, and you can see that overall, if you take a cursory look, it's basically the same. Okay? Cursory look, it's basically the same. So no matter what, you have 1C4. But if you calculate the distances between corresponding residues, there are small changes that happen. There are small changes. So there is at a micro level, there are changes. And that may generate interaction characteristics such as specificity, affinity, whatever the phenomena is. The interaction is, for most part, people believe, is strongly electrostatic. Electrostatic interactions is typically of this type. But a number of cases have been observed where the interaction between a positive charge, such as a lysine and a sulfate of a gag, can actually be hydrogen bonding. And whenever there is hydrogen bonding, there are chances of engineering specificity. This is perhaps the best. This is perhaps the best in terms of engineering specificity. So if we are able to utilize one sulfate, and in antithrombin this is actually the case. So if we are able to utilize one sulfate that recognizes an arginine such that the bond angle here is 180 degrees, this has very high hydrogen bonding characteristic, and that generates specificity. Okay. Very, very few GAG protein interactions have this sort of an arrangement. Very few. Which is the reason, for most part, people believe that GAG binding proteins interact with GAGs in a fairly non-specific electrostatic manner. Can I go forward? Yeah? Okay, here's a paper that Neha published some time ago. It's a review article, and I know it's cut off here, but in essence what I wanted to show is several different proteins, a good compilation, and you can see that GAGs, and this is primarily for heparin and heparan sulfate fragments. So you can see that the size, there is a considerable variation in terms of size. Sizes as large as 12 to 14 mers have been suggested to be important. Now we don't really know whether all of that is really needed, because that work has not really been done. The affinity is 6 nanomolar, for example. Okay, so that's pretty strong. Some of the affinities are micromolar or high micromolar. Okay, the high affinity primarily arises from multiple ion pair formation, interaction, and this is fairly easy to identify. You can actually do an experiment in which you measure the affinity of binding versus the ionic strength of the medium or salt concentration. And when we do that, for most part, the double log plot is a linear relationship according to this particular equation, which is derived from a very simple protein polyelectrolyte theory. And we can actually calculate the slope. The slope of this line corresponds to Z times psi. And psi is a parameter that relates to how well sodium is lost upon binding. But Z is a number of ion pairs that are actually formed. So you can actually find how many ion pairs are formed. And you can correlate that with how a molecule fits into the binding site. So here, for example, is the heparin binding site. The antisaccharide binding site in one of the highly charged molecules is binding. And you can, and this is simply modeling exercise, you can find out as to how, which sulfate is actually involved in the ion pair formation, and which amino acid residue is involved in ion pair formation. So you can study this from both sides. Okay, so the nature of interaction, as I have said multiple times, few interactions have found to be specific. The one that is considered as a prototypic interaction, which is specific, is antithrombin heparin pentasaccharide interaction. Other interactions such as heparin cofactor 2, dermatan sulfate, and HSV glycoprotein D heparin octosaccharide. There is a specific octosaccharide that is involved in recognition of HSV glycoprotein D, which may be specific. A lot of work needs to be done, but there are several others that have been suggested to be fairly specific interaction. But a lot more work is really needed to figure out whether this is really specific. Overall, it has been thought that the interactions arise because of sequence of amino acid residues in the protein that recognize a Gag chain, especially heparin chain. And this is a theory that was proposed in 1989, I believe. 1989. And that is usually called as the Cardin-Weintraub model, in which this particular sequence, XBB, XBX, or XBBB, XXBX, where B is a basic residue and X is any residue, hydropathic residue, is important. So it is this sequence that BBBB, and if you really figure out, most of the time this sequence is a helix, most of the time. So what happens is that these B's actually come on one face of the helix, and that's how they facilitate binding. Because heparin is a helix, for example, or short chain is a helix, doesn't necessarily mean that it's helix all throughout. A short chain is a helix, and then you have a amino acid peptide helix, and they interact together. Well, many proteins do not follow this particular simple sequence arrangement. So later on, Bob Linhart presented an even bigger sequence with a turn, a requirement for a turn. T stands for a turn, that you have to have a turn in order for it to be recognized. Well, there are some other proteins that don't follow this. It's overall, most probably, three-dimensional arrangement is really, really important. Not only that, the hydrogen bonding capability is probably important. So 435 proteins are known, are primarily, have been suggested, are primarily on the basis of what this would be. But you have to remember that although you might have something like that, that doesn't mean that it actually generates a physiological response. And that is the key, okay, identifying proteins that would do a physiological response. All right, this is something that we talked about yesterday, so I think I'll skip it. Oh yeah, I wanted to show this to you yesterday, okay? And I thought that I had it in yesterday's session, but here it is. So this is our heparin, okay? And this is the pentasaccharide binding site, and that's how antithrombin binds. For example, this is antithrombin. Thrombin binds here, and then it walks across, okay? But what if thrombin binds in an opposite direction? The whole thing will fall apart. That really means it has to go out, turn around, and bind again. And therefore, it takes a lot of time to figure out the right binding geometry.
I've been discussing this mechanism of crawling and hopping. So are there any studies, evidences for this kind of hopping or
This kind of hopping,
movement on the chain?
I wish we could see it live.
But how did this mechanism come into?
Because all the data subscribe to this logic, that the acceleration that is observed arises because of the three-dimensional movement of thrombin being restricted, now you have a template. So it becomes two-dimensional, and two-dimensional process occurs faster. So it's logical to come up with this kind of a mechanism.
You still cannot discard the mechanism that it's, it can come and directly sit next to it.
It will happen, absolutely, it will happen. But you think about the probability of finding a particular site or one particular site here versus 100 sites. What is the probability? A molecule will get into one of any site that is available, which is why nature has engineered the interaction in a fairly non-specific manner. What I'm trying to convey to you is that specificity is important as well as non-specificity is important. So you really look at the power that nature has come up with. It has used non-specificity. Okay, I'll give you one of the things that I did mention, for example, HSV. Right? All viruses that have, all enveloped viruses that have these glycoproteins, they recognize the heparan sulfate which is floating on cells. Why? Because these heparan sulfates are floating on cells like that, with all those negative charges. The virus have enveloped glycoproteins that have positive charge. So, rather than finding directly the cell surface and penetrating the cell surface, they would recognize what is floating around. And once it is captured, the virus can actually crawl into and is brought closer. So it's a way to enhance infection, for example, right? So you don't have to agree with this, okay? Yeah.
One question from this one.
Yeah.
Suppose if you have two antithrombin binding sites are next to each other, say a pentasaccharide.
You have a pentasaccharide and a pentasaccharide.
Another pentasaccharide, maybe one or two residues apart. Then, how do you think the thrombin has to come from two different sides? Or because there's no room, because
you
said that antithrombin is here, heparin is coming this way, and thrombin is coming here binding. So I'm just curious to know.
So you have to remember that this pentasaccharide is a directional sequence. It is not a non-directional sequence. Okay, it's a directional sequence. So this can be arranged in one specific manner. Now, if this is the direction in which antithrombin has to bind so that thrombin can come from here, then if thrombin comes from here, it is meaningless. It's not going to work.
That's what I'm curious. Yeah.
Okay, it's clearly not going to work. So if you have two and they are oppositely oriented, it will be completely inhibited. Because antithrombin is present in blood at a significantly high proportion. So you might have on the same heparin chain, you might have both sites occupied. And if they are opposite to each other, you will have a head-to-head or a tail-to-tail, whatever that you want to call, interaction, and that will be non-productive. So you'll have non-productive interaction, and that will go down.
So when you have two antithrombin binding sites, then it's not as good as having one antithrombin binding site.
It may not. It's not necessarily that it's not good. If, for example, this is the binding site, okay, and this is the binding site, it's not going to be very effective. But if this is the binding site and this is the binding site, and thrombin can come from both sides, it will be very good.
Okay.
Right? I don't know whether we can engineer that kind of thing. It's really extremely difficult. If you can do it, could be great. Go ahead.
I have a question. Prem has pointed out this hopping experiment and this kind of mechanism we can see inside the cell, this walking inside the cell, this store transport. So what if there is ZFP, like green fluorescence protein engineered antithrombin, and then you can probably see it?
You can probably see it. You can probably see it. You can probably see it as long as you slow down the rate at which molecules can move around. The rate at which, remember, we are talking about an acceleration of 1,000-fold. So you have to engineer conditions where you can actually see, for example, the two molecules being brought together either by quenching or by enhancement in whatever fluorescence or anisotropy or things of that sort, under kinetic control. You have to bring it down to such a level so that you can see it.
But the DNA phenomena binding proteins transcription factor sort of thing is also
available.
Similar, and they experimentally find that whether it's hop or what happens at two-dimensional walking or something.
Sure, sure. Yeah, absolutely. I mean, it's not that it's not possible, it's just that we haven't reached that level or urgency to find it, right? If the mechanism is going to really enhance the application, sure, it's something that people will spend a lot of time on, right? Okay, can I go forward? All right, this is the last slide, and I think I'll give you a break after this. So I have presented this at several of the meetings, so you all know it, but in essence, this is one of the places where we are superimposing two proteins, two serpents that are very similar. On a three-dimensional level, these serpents are very similar. And what typically happens is that the way in which these recognize, although they are similar, they bind the same binding site, the way in which they recognize the same enzyme, thrombin, is completely different. This is thrombin, this is thrombin. This is thrombin binding to antithrombin. This is thrombin binding to heparin cofactor 2. Right? So, same thing, and yet you have a completely different geometry. Similar proteins, heparin cofactor 2, and yet you have a completely different geometry. So there is a lot of scope to really come up with molecules that specifically recognize heparin cofactor 2 and specifically recognize antithrombin. Should be theoretically possible. Okay. Yeah.
So if it, the heparin chains are what, 200 disaccharides long typically?
Could be 50, yeah. I mean, 200 is pretty long. 80 is perhaps the max, something of that sort.
So how many of those would be the unique pentasaccharide sequences? What is the...
Pentasaccharide is present in 1/3 of heparin chains. So if you take 3 chains, there is 1 pentasaccharide. Now, Kubi can engineer 3, 4, 5 saccharides per chain. That's possible, because he has this in vitro system. Sure. And that will be really interesting to figure out. Absolutely.
The single chain can have a lot of 83 complex with a pentasaccharide, right? Then if you take a 50 disaccharide long chain, then each chain can have at least 3 antithromins bound to it at any time.
Yeah. So if you are saying, well, pentasaccharide is already a drug. Heparin pentasaccharide is a drug. Heparin pentasaccharide has 1,3-O-sulfate group. It binds antithrombin really well. Rather than, and heparin pentasaccharide is difficult to make, but we have a polymeric heparin in which we can engineer 3-O-sulfation. So how about converting, for most part, nearly all sequences, take heparin, nearly all sequences into 3-O-sulfate containing sequences. So you have a pentasaccharide, active heparin pentasaccharide all throughout, multiple copies of the same sequence. Yeah, you might get activity and utilize only factor 10A directing influence, which we have seen. You might really have a very, very highly active molecule. The question is, do we want that? Do we want the activity of sub-nanomolar? Because pentasaccharide is also inducing bleeding. So what do you think? So yeah, we can do it. If we do it, we might actually, not necessary that we'll end up with a best result. Not necessary. Any other question? Okay. So let's take a 5-minute break. I wanted to give you a break at 8:55, but it's 9:10. So, and then we'll come back and do techniques. And that's going to be a fairly simple
slides for them.
Oh. This is his, right?
Yes.
So we'll distribute at 10.
Okay.
print lecture notes.